Hi everyone. Uh, Good afternoon. I'm Kiet Lei, Technical Support Officer at First Base. Okay, so let us start with the today's webinar with the title "Unlock the DNA Codes of Microbes." Okay, so in this webinar, we'll go through an introduction on what is DNA barcoding, and then the basic workflow of DNA barcoding. Then we'll have some intro on our DNA barcoding kit and the technical insight, and then follow with a short conclusion. Also, there will be some good news to you in at the end of the webinar. So do stay tuned with us until the end of this webinar. Okay. So, okay, let's start with the introduction. Okay, so why we use barcode? Okay, uh, in our daily work, when you go to supermarket to purchase some items at the counter, the cashier will scan the barcode of the item, right? And then you'll find that, okay, date, and then all the information regarding the items like the manufacturer, the expiry date, the price of the item will all input inside the computer of the cashier. So, okay, this make your purchase fast and easy. So, what is barcode give you is the information of the item, right? But in biology, why we do DNA barcoding? Okay, because it will just like the same as the barcode, so it will give you the information of your microbes. Okay, so by using a short gene region from the genome, or we can call it as a conserved gene, it can ease the identification, discrimination, and classification of an organism. So this conserved gene is usually short, easy to amplify, and they has a reasonable variable between organisms. So for example, the conserved gene used for DNA barcoding of bacteria is 16S rRNA gene. For fungi is ITS. Algae is 18S rRNA. For animal, we use COI. And for plant, there is two. One is RBCL, another one is McK. All this is the conserved gene use. Okay. Okay. So basically, there is two methods used for identify microbes, which is by DNA barcoding, and another one is through biochemical tests. Okay, so uh, each different species actually of microbe, they has different and unique sequence of DNA, so that they will encode for different and unique set of protein, which can catalyze different biochemical reaction. Thus, these two methods is used to identify a microbes. And here we will have a brief comparison on these two methods. So for both systems, it is important that the microbe must be culturable, and it should be a pure single isolate. Okay. Then what you need is only a basic laboratory skill for to carry out these two methods, and the time needed is quite short. You will know what is the species of your sample in just a few days or by a week time. Okay, so the difference of these two methods is DNA barcoding has a more higher efficiency than the biochemical test. Okay, so DNA barcoding, the efficiency is 90.6%, and biochemical is 68.3%. Okay, so also the DNA barcoding is used for novel species, which is unknown species, because you can actually compare the similarity of the unknown species with a huge bunch of known species in a database. But for biochemical tests, you will need to have a hint of the biochemical profile of the species, then only you know which test to be carried out. Okay, so both species is actually both system is actually accurate up to genus and species level. So for DNA barcoding, if you compare in a standard database like NCBI BLAST-N, if you give you accurate up to genus level, but if you compare with a more well curated or a more specific database like the rRNA database with a megablast, then it can be accurate up to maybe the species level. Okay, it will depends on the comparison or the similarity of the sequence from your sample with the database. Okay, so for biochemical method, there is possibility of misidentification because biochemical method is depending on the reaction of protein in the species, and the amount of protein produced can always be affected by the growth environment of the microbe. So okay, and lastly is just for your information. Okay, so the DNA barcoding system will usually come in kit, and the biochemical test system usually come in 96 well plate or strip media. Okay, then okay, we go into the workflow for DNA barcoding. So what is the workflow? In DNA barcoding, it is involved only 4 steps, 4 major steps. Okay, so the first one is the genomic DNA extraction or gDNA extraction. Then after you determine your gDNA, you will need to amplify the conserved gene with PCR application. So after PCR, then you need to perform a sequencing to get the sequence of the conserved gene, and then it goes into result analysis. Okay, so the first step is gDNA extraction. Okay, so for gDNA extraction, basically there is 2 methods: a solution phase extraction and a solid phase extraction. Okay, but for these 2 method, what you need is a commercial kit or extraction buffer only. So for gDNA extraction, usually there is 3 steps needed. It involves the lysis of the cell, then to get the component inside the cell, then you will need to separate the DNA from the cell debris, and then wash or purify the DNA. After that, lastly, we will need to elute the DNA so that you can determine a clean and pure gDNA. Okay, so after you determine the pure gDNA, we will need to carry out a PCR. Okay, PCR, a polymerase chain reaction, is to amplify and also create multiple copy of the conserved gene by using gene-specific primers. So what you need is also very simple: the polymerase, the DNTP, the PCR buffer, the gene-specific primers, and also template, which is the genomic DNA from your gDNA extraction. And lastly, you need the thermocycler for to run all this reaction. Okay, so after PCR, it is very important that you need to verify your PCR product to check whether there is amplification, whether the size is correct, by using agarose gel electrophoresis. Okay, so after you determine your PCR product, what you need to do. Okay, so before sequencing, you will certainly need to purify your PCR product. Then we'll go into sequencing so that you can determine the sequence of the conserved gene of the microbe. Okay, so usually sequencing is carried out by using Sanger sequencing method, which using strand terminator chemistry. Okay, so what you need is about the same in PCR, except for sequencing, it will only use one primer in one reaction. And also there is the DNTP, which is come from the strand terminator chemistry. Okay, so what is so special with this DDNTP? Okay, okay. In sequencing, in PCR, okay, in PCR, after the denaturation of the DNA, the forward and reverse primer, they will bind to the template. Then the DNTP, they will come and extend the fragment. Okay, how it extend is there will be nucleotide go into and have the reaction with this OH group of the 3' of the DNTP, so it will have the elongation. Okay, but in sequencing, okay, the DNTP is carrying a hydrogen group at the 3' end. So it will stop the extension of the fragment. So the in sequencing, everything will be the same. There will be denaturation, binding of the primer, then there will be extension of the fragment with the DNTP. Okay, until the when the DNTP come to the extending fragment, then the extension will be stopped. And because this DDNTP, they will carry a fluorescent. So later they will give a signal when you run in the genetic analyzer. Okay, so before you run in genetic analyzer, you will certainly need a post-sequencing cleanup for the extended fragment so that you can clean up all the extra chemistry of the sequencing, so to avoid them from affect the result in when you run in genetic analyzer. So after this, we'll need to mix with the hydroformamide and run in genetic analyzer. And also there will be a software used to analyze and call out the bases in the sequence for you so that you will determine what is the sequence of your conserved gene. Okay. And okay, lastly is the result analysis. So this is the most important part. So when you determine the sequencing result, usually there is a forward and reverse direction. Then what you need to do is trimming of the sequence and then perform the alignment. So trimming and alignment can be carried out by using some specific software. And then after alignment of the forward and reverse sequence, you should determine a full sequence of your conserved gene. Then you can use that sequence to blast, so that to blast at a specific database so that you can compare and then you know the similarity of the sequence of your sample to the sequence in the database. Okay, so okay, that's all from my part. So I will pass this webinar to my colleague, Miss Jenny. She will tell you more on DNA barcoding kit. Okay, please wait a moment.
Hi, good day to every participant that joined our webinar. So my name is Jenny. So I'm happy to share the part 2 of how we unlock the DNA codes of microbes. So after you all have listened the our Miss Gatley about the fundamental of the DNA barcoding, I think you have more ideas how actually the DNA barcoding working. So today, uh, First Base actually would like to take this opportunity to introduce our first developed DNA barcoding kit series in the market. So for your information, we have enriched our 10 years DNA barcoding experience into this unique kit in one kit. Okay, so perhaps a lot of customers know First Base actually do provide DNA barcoding service. So I think from the past experience, a lot of projects we have been done before for our customer. Therefore, we have really earned a lot of experience from the past 10 years experience. So what is um, the benefit or what is the advantages of this kit? Okay, we can uh, in the market, maybe there is other DNA barcoding kit, but for First Base, we can ensure that the kit that we develop actually is in a good quality, robust, and carefree. Carefree because I think uh, most of you know First Base is the Sanger sequencing manufacturer. That's why we have a very good experience on the DNA Sanger sequencing part. That's why by sending the sample to First Base, definitely you don't need to worry about the result of service. Okay, we put everything all in one so that we can ease our customer to to get to get the result easily. So okay, if you refer to the slide, okay, ta-da, this is the barcoding kit that we actually did develop for our customer. So you can see from the photo, there's two kind of packaging that we have developed. One actually is inclusive of extraction kit, which is that we will include the extraction part with all the material or content for our customer to start with of A to the PCR step. So another one will be the PCR kit packaging. This one is without the extraction kit. However, these two kits actually prepare in same quantity, which is 50 preps packaging. So similar with the fungal DNA barcoding kit, we developed this toolkit with the same design and same packaging. So if you're interested to know more about this, feel free to let our you may contact your country distributor or refer to our international sales manager, Miss Rose. Okay. So let's see what is the content is in the First Base DNA barcoding kit. Okay, as you can see from the slide, for the green part, actually this is the complete kit that I mentioned just now. which is we include the extraction and the PCR reagent inside in this kit. Okay, basically, customer just need to send us the agar gel, or cell pellet to kickstart this kit if you want to use it. So for this series of complete kit, we have provided bacteria and fungal one, which is 16S gene and ITS gene to target these two organism of the studies. Okay. As for the time that you need to use, since this is included the extraction, so you need either another less than 1 hour, less than 2 hours, or another, maybe some of the protocol you need overnight extraction, and plus another 45 minutes for standard for the PCR part. For the kit content itself, for the extraction kit, complete DNA barcoding kit, you can get the lysis buffer, TE buffer, proteinase K solution, and also others like primer mix, enzyme, PCR buffer, positive control, and sequencing primers as well. But take note about this. Okay, for the primer mix, actually this primer set is specifically used in the PCR work. So if after the PCR you want to send to first base for continue to doing the sequencing, then you need to aliquot the sequencing primer for us to proceed accordingly. And the storage and the shipping for this kit actually is in -20 degree. For the cost of the kit, it costs only like USD 10 per preps. If you compare with the barcoding service that we offer, actually this is more cost effective. But if you need further clarification about how to save your money, or how do you get more details about this packaging, you may always refer to the distributor and our sales manager. Okay, for the other part, DNA barcoding PCR kit, most of the content is the same, just that due to its exclude of the extraction part, the kit that you can see only is ready in the PCR reagent. So the starting material, this one for the PCR kit is from the gDNA. And the PCR, the time that you need, because you exclude the extraction part, so what you need to have is only spend 45 minutes for the PCR, that's all. And the kit content, you just have the rest of the material, which is 16S primer mix or fungal primer mix if you are doing fungal study, enzyme, PCR buffer, positive control, and sequencing primer, same as the DNA barcoding complete kit. So storage and shipping will be the same, -20, and the cost will be slightly lower, which is 7.5 USD per prep. Okay. So since we are having a fundamental just now about the information, the technical insight. So now what I would like to share is about the kit, about the sharing of the kit. Okay, so you can see from here, first base DNA barcoding, actually we are using the solution phase extraction method. So this is a method that commonly used lah. Okay, for the proteinase K, we also included inside. This proteinase K actually is to help to digest the protein from the cell, which means it will also digest the nucleus and protect the gDNA from the degradation. Okay, and for the TE buffer that we ready in the kit, the function of the TE actually is to protect our gDNA from degrade for long-term storage. And another important point that I would like to highlight is the sample that from agar from the sample they use for the gDNA extraction must be pure isolate and culture in a agar culture, or either the cell pellet in a liquid culture. Okay, this part actually is just a very simple step. It involves lysis step, washing step, and elution step only. So the total time that we need to spend is around 30 minutes only. Okay, for the after the extraction, definitely we need to go for the PCR. Okay, for the PCR, with our experience in DNA barcoding, we do have a very well developed protocol. They are a specific primers, which is we mentioned just now, that it actually will be given for both bacteria and fungi barcoding kits. For the bacteria barcoding kit, we are using 16S primer mix, and then for the fungal barcoding kit, we are using the ITS primer mix. And not forget that we also provide the positive control inside the kit. This is to make sure the user PCR actually is running well during the process. Okay, most of the PCR, after we have do the amplification, the next step should be check with the gel electrophoresis. So after this, okay, if you refer to the screen that I display here, actually we should expect the target PCR product with the size of 1,500 base pair for the bacterial DNA barcoding. Okay, and for the fungal DNA barcoding, the size of it, the expected size of the target actually is around 700 base pair. If you see the gel photo from here, you can see there's a column which is labeled as the negative control. So don't forget, we always recommend user to include the negative control by using water as a template. This is to counter check whether the sample actually is qualified to proceed to the next step or not. Okay. Okay. After the PCR, this is something that very familiar for most of the molecular students, I think. So here in this chart, actually is the flow, or is a simple chart that we want to highlight to our customer. What is the process of the flow of the sequencing? So you can see from here, the PCR products that ready actually need to be purified before you go into the cycle sequencing. Okay, first of all, purification, you can see from the red column, this is the part that we need to do so that we can remove the excess primer and salt. So in this kit, actually customer no need to do another purification before you send to us, because in our first base facility, we can offer another service called sequencing plus service. This plus service actually is another extra step that we can help customer to clean up the sample. So by adding on a little bit of cost, actually you can get everything done. After that, after the purification, the following step will be the cycle sequencing. Okay, what is the cycle sequencing involving? So cycle sequencing actually we involve the chemistry that used in our sequencing, which is big dye terminated version 3.1 cycle sequencing kit. Okay, we also use the forward and reverse sequencing primer that provided in the kit that mentioned in the content just now to proceed accordingly. Okay, So after the sequencing reaction, means the cycle, so we will go through another step, which is called post-sequencing purification. This is just to remove the excess dye, DDNTP. After that, the final step will be the loading in the capillary of electrophoresis. So in first place, we are using the high throughput genetic analyzer, which is called ABI 3730XL. So and lastly, we will use the base caller software, which can give you the qualitative value of each base's call. So the final part is quite important for the bioanalysis. Okay. So you can see from this slide, okay, whenever you get the sequencing result after the PCR, normally you will get the electrophoresis that you can see from here. Okay, the quality value actually is a very important role in the result analysis. It lets you know whether the sequence is confident or reliable or not to get into another step, which is the alignment or the blast result. Okay, we are using the ABI DNA sequencing analysis software version 5.2 with a KB based caller for all our sequencing analysis. So as you can see here, if you open the software that we are using called Sequence Scanner, if you see the QE is in blue bar color, which is QE 40 or above, then you can trust this sequence. It is a reliable sequence that can be used in the next analysis. Okay, this is the following step that is most important for the DNA barcoding user. Okay, so first of all, after you have get all the result, sequencing result from the manufacturer, in this case is first base. So first of the important point you need to remember is you need to trim off all the unnecessary sequence. Okay, don't just dump all the sequence into the software and proceed with the alignment directly, because you might find that if the sequence is unreliable, actually it will give you some false result. So analyze the sequencing result with the sequence scanner software, which is provided by ABI. It's a free software. You may download it from the result email that we provided. Okay, by just register it, very simple, and then choose the CRL, which is the good QE bar length sequence that we can see from the electrophoregram. Then after that, you may just perform an alignment for both forward and reverse result to get the full length of the gene sequence before you blast in NCBI or any relevant database. Okay, this is based on your own preference. Okay, otherwise NCBIs will be the common website that normally we are using to check the genus of all the species. Okay, after we had mentioned the idea of the barcoding kit fundamental, so you might need to know more about this, how actually we develop this kit. So hereby I would like to share about some insight about the DNA sequencing result for DNA barcoding kit and the fungal DNA barcoding kit. What's the difference? Excuse me. For the bacterial DNA barcoding, the PCR primer actually will give you approximately 1.5 kb target size. Okay, so however, you will expect shorter read length in your sequencing. Okay, why? This is because the sequencing primer actually were designed as internal primers for 16S RNA gene which is the bacteria gene. Okay, in a sequencing result, there is an expected loss of the sequence in the front part after after the priming of the primer and sequencing. It will read up to around 1 kb only. Okay, you can see from here. So usually there are some loss from this side. Okay, eventually the maximum length that you can get if your sample is pretty well, concentration is very good, then you may only can get 1000 base pair only. Okay, so if you are using the PCR primer to sequencing, if you are using the PCR primer, we assume that you will get a region of sequence to be aligned like this. Okay, so it's a small part of in the middle that you can see we highlight in the orange box. Okay, but you will not able to determine the full sequence of the PCR product. Okay, you can see there's a loss at the beginning and at the end of the part. So, however, if you see, if by using the internal primer of 16S, okay, which is the sequencing primer for read both towards each end, you may able to get a small region aligned like this. Okay, quite small. Okay, but anyhow, you will get the loss sequence of the front part of the sequencing result covered by the opposite side of the reaction, which is like this. Okay, so in this picture, it's clearly mentioned about that you will get the full length of your PCR product from the sequencing result. And how about the fungal DNA barcoding kit? So for the fungal DNA barcoding kit, the PCR primer will give you approximately 700 base pair, shorter than the bacterial barcoding kit. Okay, and the sequence will also give you about the same length, except there is a loss of the sequence after the priming site, which is also can cover covered by the sequence result from the opposite side. This is because the PCR is short, the PCR product is short, and sequencing read length can usually read up to 1 kb. So one reaction can actually cover nearby all sequence, and you will find stop point in the end of the result. Therefore, from the alignment of both results like this, okay, you will get a full length of the sequence of your PCR product. Okay, so let's come to another slide, which is you can see there's some of the bacterial names that you can see from the list. So from the table, actually it shows that the microbes this is all the microbes that has been validated in our lab by using this kit before in the past 10 years, like we mentioned just now. So we have a lot of confidence on this. You may check also all the listed microbes in our manual. So whenever you purchase a complete kit or the PCR kit from us, definitely you will be given all these validated listed bacteria DNA for your reference. Okay, so you always can refer this either from our website first. If you are not, if you are planning to buy our kit, but you are not sure whether this has been validated before, you always can refer to our First Base website. Okay, you can download the manual and then you can get the list from us. Okay, well, since we talk a lot about this, the advantages or the technique of the barcoding kit, who is the user or who is the researcher actually can get benefit from this barcoding kit? As you can see from here, barcoding kit actually is pretty useful for most of the agriculture user, environmental or biodiversity user. or any food tech or food science user. So most of the participant or most of the researchers that are involved in this study, actually you need to do a lot of identification or a lot of verification study. So in this case, you actually can try this barcoding kit, and you will see it is easy to use, and it will give you a pretty reliable result. Okay, let's come to the end of the, it's not the end, almost. Okay, we have good news for all the participants that attend our webinar. So from the past 10 years, we share our experience before with our customer, and now we concentrate or we enrich the experience in this DNA barcoding service. But some of the customer might wondering, but I'm not doing the bacteria study, I'm not doing the fungal study, so this is something that is not related to me. Oh, if this is the thought, so you need to correct it, because first base does not only provide bacteria or fungal DNA barcoding kit, we also can customize the DNA barcoding kit just for your study specifically. So in case you are not doing the bacteria or fungal kit, actually you still can contact to our contact first base in order for us to help you to design the kit specifically. So what you need to do is you can contact your country distributor, or you can direct contact our first base international sales manager directly. We will definitely give you a pretty satisfied answer. Okay, other than that, maybe some of the customer also doing something similar, but they are not using the Sanger sequencing method to perform this barcoding kit. So let's say if you have a water or soil sample with the mixed isolate and with a loss of microbe inside, okay, how you need to do actually, you can consider to identify all these microbes by using our Amplicon service, sequencing service. This might be another option for you. You can see from here. So this is an NGS service also provided by First Base. So in case you need it, you can always refer to us. For another further verification that for your optional barcoding study, we also have some other MBS service that always can support you all. Just feel free to contact us if you have any inquiry. Okay, another custom service that we provide also, not only to develop a custom kit, we also can custom the DNA barcoding service specifically for our customer. So First Base offer this work plan. This work plan can be reviewed and accepted by the customer first. Okay, if you confirm and you are satisfied with the plan, then we can help you to proceed accordingly. Okay, what you need to do is you may just provide the primer design for the custom DNA barcoding. So, but there is a terms and condition. All the custom DNA barcoding will require minimum of 5 samples. Yeah, at least 5 samples. So if more than that, no problem, but it shouldn't be less than that. Okay, let's come to a conclusion. So DNA barcoding is not only used for identification purpose, but also serve to authenticate the biological materials. So in this First Base DNA barcoding kit series, we kickstart with two gene, which is bacteria and fungal barcoding. Okay, and the packaging will be provided according to whether you need inclusive the extraction part or exclusive the extraction part. So if you need further information or clarification, okay, again, feel free to contact to our distributor and first base directly. Other than that, this barcoding kit actually is a door opener for us to customize all the DNA barcoding kits that you need. Okay, we can help you to design the kit or either service as well if you need it. Okay, and not forget to mention, in this launching period, if you commit or if you purchase any kit from First Base, actually you can get a 10 sequencing plus reaction service for free, or 10 reaction service actually is a very attractive bonus. Okay, so make sure that you don't miss this, because first come first serve. So we hope that you can quickly book this from your distributor or from us directly, okay, to get this good deal. Okay, again, to make a final conclusion, okay, these barcoding kits, actually, if you can see from here, we provide the best quality that we could. We give robust services, and we make sure the kids or user experience, when you use it, you no need to worry too much, because we have a good experience in Sanger sequencing. Just pass the sample to us, then we will definitely help you to settle everything. So everything is all in one. Okay. And for this Friday, yes, this Friday, you can see from the PowerPoint here. Okay, we have a Facebook Live or YouTube Live in the same time. In Malaysia time, actually is 3 p.m. Okay, we will have a talk, or if we have a small sharing about the unlocked DNA codes of microbes as well. So stay tuned with us. I think you will get a very attractive gift if you stay until the end of the FB or YouTube live. So don't forget about it. And we will have our international sales manager to see you on Friday. Okay, Miss Rose will be the in charge. Not in charge, but the person will be share all the good news to you. Okay, we hope to see you. Okay, thank you for that.